Hi everyone, today I'm going to talk about the olfactory system. The basic function of the olfactory system is to detect odors. Chemical stimuli in the form of odorants trigger depolarization of olfactory sensory neurons of the olfactory epithelium. Now this cartoon illustrates an olfactory pathway activated from the smelling of a pink rose. Now today we won't cover this whole process, but we'll focus on the area inside this green box, describing a bit of anatomy and focusing on the histology of the different types of epithelium in the olfactory system. I'll be covering olfactory sensory neuron signaling in a different tutorial. I'm Catherine Moore, the Histology Wizard. Let's start with a bit of anatomy. Here we're looking at a frontal view of the skull. Remember that the right side of the body is on the left side of your screen. Here we see the paranasal sinuses, air-filled spaces located within the bones of the skull and facial bones. They're centered on the nasal cavity and have various functions, but for olfaction, they help humidify and heat inhaled air. There are four sets of paired sinuses: the frontal sinuses, the ethmoid sinus, which opens beneath the superior concha bone, the maxillary sinus beneath the middle concha bone, and the sphenoid sinus, which isn't seen in this view. These cavities directly communicate with the nasal cavity, and secretions from these sinuses drain into the nasal cavity via the thin-walled ostia or holes. Now let's overlay a cartoon to highlight and narrow in on the olfactory system and the nasal cavity, and remove the rest of the skull. The nasal cavity is the most cephalic part of the respiratory tract. It communicates with the external environment via the anterior apertures, aka the nares, and the nasopharynx via the posterior apertures, or conae. Now this cavity is divided into two separate cavities by the nasal septum, and it's kept open, or patent, by a bone and cartilaginous framework. Each cavity consists of a roof, floor, medial wall, and lateral wall. Now let's take a closer look at the nasal septum. Here you see a frontal section of a developing head. Now, the shape isn't exactly like the adult form, but we can still identify structures like the nasal septum. The adult nasal septum is composed of cartilage and bone covered in mucosa. You don't really see bone at this stage, but you can see the cartilage in the center, surrounded by the lamina propria and olfactory epithelium. And if we take a closer view, we see that this is hyaline cartilage. And you can tell it's hyaline cartilage because it has that kind of shiny, glossy appearance, a low number of fibers, and chondrocytes that are forming isogynous groups or nests. Next up, we have the nasal concha or turbinates. These are long, narrow, curled shelves of bone that protrude into the nasal cavity. They look a bit like elongated seashells, which is what gave them their name. Now you can see them colored blue in this cartoon overlay. These bones have multiple functions. They're mostly lined with respiratory epithelium, they've got a lot of goblet cells, and the structures help humidify and warm our inspired air. They also are enriched with multiple temperature and pressure sensitive tissues that are linked to cranial nerve 5. These turbinates or concha also increase the surface area of the inside of the nose, and they sort of deflect and direct airflow across the mucosal surface of the inner nose. So they actually kind of propel air. In essence, they act like baffles that makes this inspired air really turbulent. And so this kind of mixes up and carries the scent molecules and wafts them towards the higher, very narrow regions of the nasal airways. And that's where our olfactory receptors are located. So let's walk through the different conchae and their associated meatus, or openings. First, we have the superior nasal concha and meatus, and that meatus drains into the sphenoid and posterior ethmoid sinuses. And this also helps modify vocal sounds, interpret smell. The middle concha and meatus drain the frontal, anterior ethmoid, and maxillary sinuses, and the inferior concha and its meatus, which helps to control airflow direction and humidify and filter air. And this meatus also contains the nasal lacrimal duct opening. Now in this image, you can see all three of these conchae labeled. And in this H&E frontal section, we can actually identify the bone of one of the concha covered in olfactory mucosa, which is lamina propria and olfactory epithelium, and we're going to look at more closely in more detail in just a few minutes. But I want to hone in at this point on the area of that superior concha and look at the mucosa, the actual olfactory epithelium. So let's take that frontal cartoon section here and look inside. First, we're going to bisect the area, and then we're going to kind of flip up this left side. And here again, you can see the nasal septum and that superior nasal concha. Now I'm going to layer in a slightly different cartoon where we've reflected the nasal septum anteriorly. Now here again, we can see that nasal septum, we can see the superior concha, and now we're adding in those olfactory neurons which detect and transmit signals that convey odor to the rest of the brain. Okay, so keeping the locations of these structures in mind, let's look at this from a slightly different angle. Here's another cartoon. This is a mid-sagittal section of the nasal cavity. And within this nasal cavity, we have three big regions: nasal vestibule, the respiratory region, and the olfactory region. So I'm going to review each area and then look at some histology. So we're going to start with the vestibule. This word means antechamber. So the vestibule is basically the lobby of the nose, where the air enters the body. Not surprisingly, since it's continuous with the outside of the body and with our skin, much of the vestibule is lined by squamous epithelium. So we have a basal cell layer, several layers of squamous cells that get flatter as we go towards the luminal surface. Essentially, this is skin, thin skin, keratinized stratified squamous epithelium. It has a dermis underlying it with sebaceous and sweat glands. And similar to skin, the function of this lining protects the underlying tissues from harmful atmospheric agents. It also has short, stiff little hairs called vibrissae, and these help filter dust and other particles. Now, the vestibule itself is supported by hyaline cartilage and bone. The covering of the second half of the vestibule is respiratory epithelium, pseudostratified ciliated columnar epithelium. Next, we see our nasal cavity, those concha again, and this pink patch, which represents the area of the olfactory epithelium that contains the olfactory receptor neurons. And finally, this yellow structure is the olfactory bulb. This is the location in the brain where the axons of the olfactory receptor neurons synapse to transmit signals to the higher brain centers for odor detection. So for the rest of our time, we're going to focus on the histology of three of the major components of the olfactory system: the respiratory epithelium of the nasal cavity, the olfactory epithelium of the nasal cavity, and finally, we'll touch briefly on the olfactory bulb. The actual signal transduction of olfaction and more details will be covered in a separate tutorial. Now note here that the olfactory epithelia is shown as we typically view this kind of epithelium, and our basal cell layer, which we'll talk about, is actually facing up. But we're going to start with respiratory epithelium. Now, the respiratory region of the nasal cavity functions to humidify, warm, filter, protect, and eliminate debris from the air. As air traverses the nasal cavity, it warms up to about body temperature, and it actually reaches around 100% humidity. The vascular or neurovascular supply of this region is important for this function. It regulates nasal airflow, it controls blood volume in the erectile tissue on the turbinates and septum. So hopefully you'll recall that respiratory epithelium, or pseudostratified ciliated columnar epithelium, has three major cell types. And again, this epithelium covers most of the nasal cavity and lines the paranasal sinuses. So first we have our basal cells that form the deepest layer next to the lamina propria, our ciliated cells, and goblet cells. Particles that get past that nasal vestibule become trapped in the mucosa of the nasal cavity, and that mucociliary system with goblet cells and ciliated cells helps to get rid of these particles. The cilia sweep particles into the nasal pharynx for further expulsion, and the mucus produced by the goblet cells also forms a protective barrier to inhaled pathogens. Now here you see an H&E stained section showing respiratory epithelium. Here we can see basal cells, goblet cells, and ciliated cells. We have a second example that shows basal cells, goblet cells, and ciliated cells. Next, let's look at the lamina propria of respiratory epithelium. This bracketed region here is the lamina propria, all the way up to the bone, but not including it. It contains typical loose connective tissue. Some areas contain large venous sinuses, and this helps control airflow via sympathetic innervation. And there are a lot of seromucus glands as well. Next, let's look at olfactory epithelium. This specialized epithelium lines the roof of the nasal cavities, part of the nasal septum, and the superior, sometimes the middle, concha or turbinate bones. This is the epithelium that's responsible for odor detection and transmission of odor information via action potentials to the olfactory bulb of the brain. Now, a lot of people think that respiratory epithelium and olfactory epithelium look identical, and they are similar, but there are important histological and functional differences. In this H&E section through the nose, we see the transition from respiratory to olfactory epithelium. And if we look at higher magnification, you can see that the respiratory epithelium contains more goblet cells and is overall lower in height than the olfactory epithelium. And these differences are even more obvious at higher magnification. So let's move on to olfactory epithelium. This epithelium also contains three major cell types. Basal cells. Now, here I want to point out that the basal cells in this cartoon are on the upper part of the image, in contrast to that earlier cartoon of respiratory epithelium. For the rest of the tutorial, every olfactory epithelial image will be shown this way. Now, this is because olfactory sensory neurons, or receptor neurons, face the nasal cavity, and thus the outside of the body. So this orientation is how most images of the olfactory epithelium are shown in texts and images. Next, we have our sustentacular cells, and then finally our olfactory sensory or olfactory receptor neurons. Now let's look a little closer at each cell type. So here's our basal cell layer, and similar to a lot of basal cell layers, it's a single layer of stem cells that lies in the basal region right over the lamina propria. These cells are the stem cells of olfactory epithelium. They continuously undergo cell divisions to replace olfactory neurons and other cell types. This process is highly regulated and occurs even in adults, which make olfactory neurons one of the only populations of neurons that can be replaced in adult brain tissue. Now, this H&E section shows the basal cells with their nuclei more or less in a row right over the lamina propria, seen here at the top of the image. The next cell type is the sustentacular cell. This is actually a really cool cell. These cells are kind of partially epithelial and partially glial, and they have these interesting apically located nuclei that you can see in this H&E stained section. So here I'm pointing out nuclei of two different cells, and here you can see that these apical nuclei are lined up more or less in a row close to that apical surface. Now, these cells are non-sensory cells. They have a lot of apical microvilli that you can't really see in most images, and a lot of secretory granules. So they importantly provide physical, metabolic, secretory, absorptive, phagocytic, and a whole bunch of other supports for olfactory neurons and the olfactory epithelium in general. And interestingly, these are the cells that are highly invaded by the SARS-CoV-2 virus that causes COVID-19. And it's the infection of these cells that leads to a temporary or permanent loss of the sense of smell in many patients with COVID-19. Now, our final cell type is the olfactory sensory or olfactory receptor neuron. Now, these neurons are unusual bipolar neurons. They have an axon and many dendrites. They have thin apical dendrites, and then these dendrites have cilia that extend from these little knobs called apical knobs. And then the axons themselves, these single axons or individual axons, group together in bundles called philia that eventually form the olfactory nerve or cranial nerve 1. Now, these neurons are the only neurons in the body that directly contact the outside environment, and their cilia on these apical dendrites are the site of odorant binding detection and transduction. Now, the section seen here on the right of this next image is stained for a specific marker for olfactory neurons. And I really like this because you can really see this kind of tall, spindly look of olfactory epithelium that's very distinct from respiratory epithelium. Now, this unique structure of this neuron is what allows them to detect odorants in a process that I'll describe again in a different tutorial. Now, this H&E stained image shows the nuclei of the olfactory sensory neurons that, unlike the sustentacular cells, lie scattered throughout the epithelium. So let's review the location of the nuclei of the different cell types. First, we see the basal cell nuclei in the basal layer closest to the lamina propria. Next, we see a number of different olfactory receptor nuclei scattered throughout the epithelium, and the nuclei of those sustentacular cells that are more apically located. Now, I want to talk about a few important features of the lamina propria of the olfactory epithelium, since these contribute to our sense of smell. The lamina propria, shown here by this bracket, consists of a loose connective tissue, and there are three features I want to point out. First, we have a number of arterial and venous plexi of the lamina propria, and these are considered erectile tissue of the olfactory mucosa. These vessels allow the mucosa to both enlarge and shrink, and this is all under sympathetic innervation. Normally, this tissue is continuously stimulated by sympathetic signals, and that keeps our nasal cavity uncongested. Next, we see that in the lamina propria, a number of olfactory filia. These filia, they should look a lot to you like nervous tissue. That's what they are. These are bundles of olfactory neuron axons. There's about 20 in number on each side of the nasal cavity. Just like other axons, these are surrounded by a Schwann cell sheath, and they form the olfactory nerves that project to the olfactory bulb. Finally, the lamina propria contains a large number of glands. These are branched tubular alveolar glands. They're also given the eponym Bowman's glands. They produce mucus to moisten the olfactory epithelium and dissolve odor-containing gases. This H&E stain section shows these serous glands and some ducts, and these glands secrete this fluid that dissolves odorants. So the fluid contains this special protein called odorant binding protein that binds odorants and carries them to the surface of the cilia on these olfactory receptor neurons. Now, also importantly, the same protein, this odorant binding protein, also removes the odorants so that we can keep continually detecting new odors. The mucus also contains IgA from plasma cells for protection. Now, in this image, you can also see some olfactory filia. Here's another image showing a duct of an olfactory gland that looks like it almost extends through the epithelium to the surface, and then we can see a number of venous plexi. And this image shows all of the components I've just described, except some seromucus glands. So here we see the cilia of the olfactory receptor neurons, the sustentacular cells, the nuclei of the basal cells, the number of venous sinuses, and the olfactory filia. Now, our story isn't complete without mentioning the olfactory bulb, so let's go back to our cartoons. Recall that the olfactory bulb in this cartoon lies superior to the olfactory epithelium, which is shown here as this kind of pink square. Now, the next image shows the superior concha and the olfactory neurons. Recall that those axons of the neurons are going to penetrate through holes or foramina in the cribriform plate of the ethmoid bone. You can see that here in this cartoon, where you can see the little foramina as well. Now, those axons are going to synapse in the olfactory bulb, and signals from here will lead via the olfactory tract, eventually to the cerebrum. And in this next image, on the left, we're looking down the ventral side of the brain, and you can see the olfactory bulbs and tract. Now let's walk through the sequence one more time. The olfactory sensory neurons are primary afferent neurons, and recall that the nerve bundles from olfactory villi that form cranial nerve 1 are going to cross the cribriform plate of the ethmoid bone, as shown in this cartoon. In this cartoon, which is of the olfactory bulb, those axons make contact within the glomerulus with the dendrites of the mitral cells of the olfactory bulb, and these second-order neurons will continue the signal to the rest of the brain. Here we have an H&E stained frontal section of the head of an embryo showing from top to bottom the olfactory bulbs, the nasal septum dividing the nasal cavity, and the concha with olfactory mucosa. Now let's look a little bit closer at these layers of the olfactory bulb. This cartoon shows our layers with the olfactory neuron axons penetrating the cribriform plate. Now the main olfactory bulb, as shown in this image on the left, has a multi-layered cellular architecture, but I'm only going to talk about two of these layers. On the outer surface, shown here in blue in the image on the left, lies the glomerular layer, where direct input from the axons from those olfactory receptor neurons is received. The ends of those axons cluster in spherical structures or glomeruli. These are like balls of yarn, and they're seen here in the cartoon as pink circles. The other layer I want to mention is the mitral cell layer, shown in red in the image on the left and in pink in the image on the right. The dendrites of these cells synapse with the axons of the olfactory sensory neurons in those glomeruli, and the mitral cells in turn output to the olfactory cortex. Now let's end with some clinical pearls. Anosmia, or the lack of smell, hypoosmia, a reduced sense of smell, or dysnosmia, aberrant sense of smell. All of these can occur transiently or permanently, and there are a lot of things that can affect our sense of smell, including damage to cranial nerve 1 or tumors that compress cranial nerve 1. Now, because the olfactory neurons are exposed to the outside environment, we often have loss or damage of olfactory neurons. In addition, disease or damage to the mucosa can occur, causing sinonasal disease. And this includes anosmia caused by the SARS-CoV-2 virus, which affects olfactory mucosa, sustentacular cells, and much of the olfactory tract, including the olfactory bulb. Today, we've covered some anatomy and looked at some histology of the olfactory system. Thanks for stopping by.